Okay. Well, welcome everyone to our first science seminar for quite a while. In case you don't know me, my name's Ian Godwin, so I'm the Director of the Centre for Crop Science. And it's a great pleasure today to be able to introduce Millicent Smith, who everyone seems to call Millie, which is good. It seems to suit her better than Millicent. So Millie only joined UQ at the end of last year. So she has a joint position between QAAFI and the School of Agriculture and Food Science, where she's a lecturer in crop physiology. Millie did her PhD from the University of Sydney, and she worked on pulse crops there. She's had a number of experiences working overseas. Prior to coming to UQ, she was a postdoc at ICRISAT, the International Crop Research Centre in Hyderabad in India, where she furthered her work on pulse crops. She was working there mostly with a group that was interested in genetic improvement and genomics, but her focus has always been on the physiology of the crop, with a lot of interest actually on the nutritional quality that those legumes can give people as a food source. So it's good to be able to welcome Millie into the centre. And so just to give you a bit of housekeeping, so Millie's talk will continue on uninterrupted without questions during the time. You can see down the bottom of the Zoom link that there's a Q&A section there. It would be good if you could actually, if you have any questions, if you can put them in the Q&A component. You can use chat if you get confused, but it's best if you put them in Q&A. And so, because that's what I'll be monitoring, and I'll be asking the best questions from there. If I don't think there's any good questions, I'll just make them up myself. And I've got a list of 3 questions that I'm ready to go with. And the first one for everyone to think about is, why is chickpea called chickpea and pigeon pea called pigeon pea? No one's ever been able to tell me that. Perhaps Millie will. But we'll have those questions at the end. So without further ado, it's over to you, Millie.
Thanks, Ian. And thanks everyone for joining today. It's really nice to have the opportunity to share a bit of my research so far on pulses and set a bit of what I think we can do going forward in terms of the direction for new crops. So I told someone that I work on pulses, and they said, "Oh, so you're a heart doctor then, that's really cool." And so I thought that perhaps I should start by clarifying that this talk isn't about blood pressure, sorry to disappoint you, but it is a topic very close to my heart, and that is pulse crops. And these are a subgroup of the legume family. The term pulse refers only to the dried seed. And in Australia, we really don't typically regard soybean and peanut as pulses. They're traditionally oil seeds. So our major pulse crops are chickpea, faba bean, field pea, lentil, lupin, and mung bean. And there's also some upcoming pulses like pigeon pea. So pulses play a really important role in our crop rotations in farming systems. They are a really important disease break. They allow for better weed management, and they also improve the soil condition. They can develop symbiotic relationships with rhizobia, which means that pulse crops are capable of fixing their own nitrogen. Well, theoretically, if the plants are effectively nodulated, they really shouldn't require any additional nitrogen. And some pulse crops, particularly those that are shallow rooted and early maturing or short in duration, use a lot less soil moisture compared to cereals, and so they can leave some of that water behind in the profile for the following crops. Pulses also give farmers access to additional markets, which can spread out the risk they have for growers, and it sometimes means that pulses are considered a cash crop in some instances because they're short duration and can get a bit of money out of them if it's going well. So pulses are very good for us, and they're often, even with a cereal, offer all of the essential amino acids. So pulses have lots and lots of nutrients, particularly things like iron and zinc. And because of this really high protein content, people's protein choices are starting to drive demand in the future, hopefully more for pulse crops, but also driving demand at the moment. And with this rising trend, there's like a global alternative protein market. It's meant to be balanced, which is pretty massive. But let's face it, Australians are the biggest meat eaters in the world. We love a good steak, and so our consumption patterns, I think anyone has to stress out about that. I mean, whatever happens with the alternative protein market, it's very difficult to predict, but Australian pulse crops can play an important role in this growing space by really making the most of our expertise in ag science and by developing value chains. I think this can only be a good thing for our future pulse production. And that's because pulses are quite a long way behind cereal crops. Just pop my pointer on. So you can see cereals at the top are a fair bit below in terms of tonnes per hectare. And this is the yield over time for the globe. And there's a lower, like pulses are increasing at a lower between the two. I don't know that this trend is really all that surprising, I guess. I think it could be due to a whole range of different factors like investment. But I just thought I'd dig a bit deeper into this and think about this for two crops in Australia. And that's chickpea and wheat. Everyone loves a good fight. Let's stack them up against one another and see how they're going in terms of historic use in Australia. So we're going way, way back in time now to when both wheat and chickpea were domesticated around 10,000 years ago in the Fertile Crescent, along with other important crops like barley and lentil. And as a consequence of this domestication, chickpea and wheat have about 50% higher yields than their wild relatives. If we jump very forward in time to 1788, when around 200 years ago, Australia was invaded by the British, who brought some wheat with them. Contrastingly, chickpea was introduced almost 100 years later in 1890. So regular exports of wheat commenced in 1870, and the chickpea export market really began fully operating in around the 1980s. Commercial production commenced around this time, and the first commercial chickpea cultivar was released about 10 years prior to that. So Farrer started breeding wheat in Australia in the 1880s, and that meant that at the beginning of 1890, when there was a lot of disease and drought, it was really preventing the expansion of wheat in Australia. And so Farrer's Federation wheat variety, which was well adapted by then, could be released in the early 1900s, and wheat expansion could occur dramatically across Australia from then on. For chickpea, this production really started to increase in the 1990s, and just like wheat, but only 100 years later, the disease and drought really prevented the expansion of the crop. And luckily, New South Wales DPI and DAF had commenced some collaborative breeding efforts in the 80s, and this allowed for these new varieties that were better adapted to Australian environments and disease pressure to be released. And so in 1997, Sadiq and Sykes write in a paper that was published in the Australian Journal of Experimental Agriculture that there appears to be no major constraint to pulse production in Australia that cannot be addressed by breeders, agronomists and farmers. And I'd have to agree, but maybe add physiologists to the list of people that can help out as well. So both wheat and chickpea have been dramatically impacted by international markets. There was a first big shake-up for wheat happened in the late 60s, early 70s, and that was because of quotas and the loss of the UK wheat market, basically. And comparatively, only a couple of years ago, India placed massive tariffs on Australian imports, which sent huge shockwaves through the chickpea industry. Luckily, since that time, some exports have been able to continue to other parts of the subcontinent. So for both crops, breeding efforts have been very critical to success since the introduction, and just that wheat had a 100-year head start. So the establishment of the GRDC, industry bodies like Pulse Breeding Australia, and other companies have had massive impacts on making wheat and chickpea more suitable for Australian environments. When we get to the late 2000s, chickpea starts to overtake wheat for the first time. Pretty exciting. The genome was sequenced in 2013 compared to wheat, although I suppose I'll have to admit that it has a much larger genome, so it might have been a bit trickier. Looking ahead, the yields are really forecast for wheat and chickpea to be fairly good this year, 28.9 million tonnes for wheat and 708,000 tonnes for chickpea. I think when you reflect back on this story of the history of introduction and selection and use in Australia for both of the crops, you can see that the short amount of time for adaptation to Australian environments and the breeding and management efforts that have been made have been incredible for our growers over time. But there are still massive opportunities for both crops. Despite this 100-year head start, wheat is still impacted by the same factors as chickpea, really. So you can see that the yield in terms of tonne per hectare for Australia now, over the last 30 years, wheat is this top line here, the purple, and chickpea is the blue. And you can see there's quite a lot of variability across these environments. So if we focus in on just chickpea and mung bean yields for Queensland, the two major pulses that I could get the data for the last 30 years, and you can see there's quite a lot of variation for Queensland over this period of time. And unsurprisingly, many of these decreases in yield have been associated with major drought events in Queensland. So there was a drought very early in the 90s, the Millennium Drought, and our most recent drought, the most severe in the state's history. There's also a decline here in 2010 associated with a wet harvest. It just turns out that too much rain is also a bad thing. So there's a lot of improvement we can make in terms of our pulses for Queensland environments. So if we do want to design pulses that are better adapted to Australian environments, what are some of the key areas that we should focus on? I'm sure you all know that for any crop, yield is a consequence of the genotype, the environment, and the management strategies. And for a long time, many people thought that this was a black box. You often heard breeders saying, oh, it's just G by E. But the reality is different to that. It's very clear, thanks to researchers, including many here at QAAFI, that underlying each of these factors are physiological processes that determine yield. And these models are built on biology. It's not just made up stuff. So as a plant physiologist, I'm really interested in understanding the biological processes that govern yield and how these interact or change as a consequence of G by E by M. So what traits should we focus on for future pulse crops, and what do we really need to do to understand that biology? Well, let's start with water use efficiency. So plants are constantly balancing the acquisition of carbon with the loss of water, and we can measure water use efficiency to try and understand this balancing act a little more clearly. So water use efficiency is basically a measure of the plant's capacity to assimilate carbon or create biomass or create grain per unit of water that they lose to transpiration. So we can measure water use efficiency across a whole range of scales. At the field scale, this is indicated by the amount of water that's received or evaporated during the season, and the total yield or biomass. And we can measure this by capturing rainfall or evaporation data. At the plant scale, this is transpiration efficiency. It's the genetic component of water use efficiency. And we can measure this by weighing plants continuously over that growing period. At the leaf scale, this is intrinsic or instantaneous water use efficiency. And this is the relationship between photosynthesis and stomatal conductance or transpiration. And we can measure this using gas exchange systems like the Lycor 6800 here. But this can be pretty time consuming. So another way we can think about this is by measuring carbon isotopes. So carbon isotopes are a pretty useful proxy for assessing plant water use strategies, and to understand why, I guess I have to explain a bit of the theory behind carbon isotope discrimination. So just stick with me through this one. So basically, what happens in the process of photosynthesis, the proportion of carbon-12 to carbon-13 in the dry matter of C3 plants is less than the atmosphere, because during the process of photosynthesis, plants discriminate against carbon-13. So when the plant is under water stress, its stomates are a lot more closed, and that means that less CO2 is getting into their chloroplasts. And so they can't be so fussy anymore about the type of carbon that they fix. And so in these scenarios, we detect a high ratio of carbon-12 to carbon-13 within leaf tissue. And carbon isotope discrimination is a measure of that ratio in plant material relative to the air that supplies them with carbon dioxide. And we calculate it from this equation that was described by Farquhar and others. So the fractionation of carbon isotopes occurs during the process of photosynthesis in two major steps. So there's diffusional fractionation, termed A, basically the fractionation that occurs as CO2 is moving into the leaf. And there's also fractionation that's associated with carboxylation of CO2 into rubisco, biochemical fractionation. And there's also heterotrophic fractionation, which is fractionation that occurs generally beyond the leaf tissue. I'm not going to talk about that today, but it is interesting, and I've done a bit of work on that in the past. So Farquhar and others developed an equation to account for these dominant processes that incur fractionation. So A is termed 4.4 per mil, and B is about 24 per mil. And so then we can put in these numeric values into this equation, and you can see that carbon isotope discrimination is positively related to the ratio of intercellular CO2 concentration, how much CO2 is inside the leaf effectively, and the CO2 of the air. And so this was really nicely shown in a study by Condon and others, where they have various wheat genotypes here, and they've correlated the CI over CA against the discrimination. You can see it's a super nice relationship. Because we know that water use efficiency is negatively related to the CI over CA, and therefore carbon isotope discrimination, we can use them to effectively assess the water use efficiency of the plant. And this was demonstrated originally by Farquhar and Richards in 1984, and then effectively applied by Revetsky and Condon and others into the Australian wheat breeding program. So you can see these wheat varieties vary in terms of their water use efficiency, and that's detected in terms of carbon isotope discrimination. So I've spent a fair bit of my research career so far working in collaboration with researchers from CIAT, which is the International Centre for Tropical Agriculture. Their headquarters are based in Colombia. They're a CGIAR centre. And I worked there on common bean. It's not such an important crop in Australia, but it's a really important physiological model. And it's one of the most widely consumed pole species in the world. People all across sub-Saharan Africa and Central and South America rely on common bean as a supply of protein in their diets. So I just wanted to show this as an example for how we can use carbon isotope discrimination to delineate how genetic variation in response to stress. So you can see here, we've done a study in a small lysimeter system. We have a range of lines that are adapted to low phosphorus and drought, and we've grown them under these conditions. So there's low phosphorus and high phosphorus well watered, and low and high phosphorus water deficit. And you can see the carbon isotope discrimination from the seed, which was ground, and then we run it through an isotope ratio mass spectrometer to get the data. And you can see there's a really nice fallout for each of these treatments in terms of their carbon isotope discrimination. We really want these sorts of proxies to help us search for plants that have a nice relationship, and we can get good leaf level water use efficiency understanding. We really don't want to use carbon isotope discrimination to select for a plant that's going to sit back and keep its stomates closed all the time. And so I think it's really important to start to consider things like variation in photosynthesis and stomatoconductance, which we measured also for this trial. And so we measured stomatoconductance on the y-axis here, again for these same treatments. And this is maximum photosynthetic rate. So it's basically we have CO2 and light aren't limiting in these conditions. And the plants were measured at flowering, which is in yellow, and mid-pod fill, which is in black. So the bred lines for low phosphorus and drought adaptation are in the circles, and the commercial checks are a square and a triangle. So basically you can see that there's variation amongst the stomatoconductance in these lines that are under the well-watered conditions and also under water deficit. But there's a couple of standouts. So if you look down here, we've got relatively high photosynthetic rates and low stomatoconductance, whereas up here we have high stomatoconductance and similar, slightly higher photosynthetic rates. So we really want to use photosynthesis and stomatoconductance measures and particularly photosynthetic capacity, to try and improve yields, particularly under abiotic stress like drought. And ideally, what would have been really nice in this scenario with these lines would have been some way to screen them quickly, because it takes a while to measure all of these traits. And so this relates to another layer of water use efficiency, and I'll just show you some example now. So we want to think about how we can phenotype water use traits really efficiently. So as Ian mentioned, I spent some time last year at the International Crops Research Institute for the Semi-Arid Tropics, mouthful, ICRISAT, at their headquarters in Hyderabad, also a CGIAR centre. And I was on an Australian government fellowship there, an Endeavour Research Fellowship, and I was really lucky enough to work on a whole range of different chickpea populations and phenotype their water use. And so one was a set of near introgression lines, and these were developed by marker-assisted backcrossing of a particular genomic region, which is found on linkage group 4. And so you can see the donor parent here, ICC 4958, and it was put into some elite chickpea cultivars. And this had happened previously in 2013, some work published by Rajiv Varshney and others. And this showed that when this region was introgressed into a commercial cultivar, JG11, the introgression lines had much higher root length density and increased yields up to 25% under drought conditions, which is pretty massive. So we took another set of these introgression lines with a whole range of elite parents and phenotyped them on the LeasyScan platform. So the LeasyScan platform is a really cool platform. It helps us to look at canopy architecture traits. So it's scanning using lasers and fancy cameras, taking photos all the time, every 50 to 80 seconds. And then these photos are reconstructed and they become 3D images. And so fine scanning of all these above ground properties like 3D leaf area and angle allows for the sort of projection or prediction of canopy architecture over the plant's development. And so they're scanned on the LeasyScan under well-watered conditions for about a month. And at the end of this time, I took some leaf samples for carbon isotope discrimination. And we use this as a proxy for water use. So carbon isotope data was sampled at ICRISAT and then kindly analysed by Margaret Barber's lab at the University of Sydney. And what we found was, so this is the elite parent, and this is the introgression line with this special genomic region. And there wasn't any significant changes in carbon isotope discrimination between the parent and the introgression line. You can see that there's a whole range of water use strategies here potentially. Carbon isotope discrimination goes up in this one, goes down in the other. It's really a bit tricky to see that the genomic region is having any influence over carbon isotope discrimination here. What was super cool though is that the data from the LeasyScan showed that leaf area significantly increased in many of these lines compared to their parent. So this is an elite donor, and this is the introgression line, and you can see that it's gone up compared to the parent. And you can see a little snapshot here of some of these leaf area increases. So I thought this was really cool, but it gets better. Interestingly, the rate of leaf area increase also differed between the elite lines. I've just taken a snapshot here. This is JG11, which I showed you earlier, and these two introgression lines. You can see JG11 has a lower growth rate compared to these introgression lines that have a bit of an increased predicted leaf area growing more fast over time. And so this is really interesting. It sort of suggests that this region is involved in vigour, and that has been shown previously in the literature and also in other crops, that early vigour is a really important trait for water use because it allows for increased canopy cover, and this can reduce things like soil evaporation, which increases the water that's available in the profile for the crop later on. So the principal component analysis showed that these traits are associated with vigour are all related to one another, unsurprisingly. So there's vigour and 3D leaf area, growth rate, specific leaf area, a whole range of things there. They're all closely associated to one another. What's interesting to me is that carbon isotope discrimination was not as well correlated with these other traits, and that suggests that there's something interesting there to explore a little further, different mechanisms potentially between these two traits. I think it would be also super cool to explore these traits as well under some water stress conditions and maybe phenotype the architecture under those conditions as well. So as we just explored, water use efficiency could be used for a whole range of different traits or associated with a whole range of different things like root architecture, for example, canopy architecture, and early vigour is obviously very important. The timing of water use is related to early vigour and incredibly important as well, especially for a lot of our rain-fed crops. Stomatal conductance is something we need to consider, and also VPD, which I didn't touch on today, but it probably plays a really big role in this. It's really critical that whatever we do, we attempt to improve water use efficiency and we phenotype under both well-watered and water stress conditions. So I showed briefly earlier, there's a bit of variation in common bean, at least it seems, for photosynthetic capacity for pulse, and it's probably the same for other pulse crops. And photosynthesis is really closely coupled to yield, and so it's likely to be a really important trait for improvement. But what we're not sure about is whether we need an increase in sink strength to make the most of any efforts that can improve photosynthetic capacity. So what do I mean by this potentially? Well, here's an example from RCN, my QAAFI colleague, and this shows some of his agronomy trials that were completed across central and southern Queensland as part of a GRDC-funded pulse agronomy project. And this data here shows a really nice relationship, exactly what we expect. So in terms of dry matter and grain yield, there's a very nice linear relationship. But you might have noticed that part of the graph is missing, and that's because it gets a little bit confusing after 5,000 kilos of dry matter is produced. The whole relationship sort of falls apart a little bit. Just ignore the linear line there, and it just seems like there's just a random scatter of plots. And what it's showing is that an increase in dry matter isn't really leading to any proportional increase in yield like we'd expect. So it's probably a result of environment and a whole range of other factors, but it kind of indicates some sort of limitation in partitioning to the reproductive tissue. There's obviously carbon there. The plant has a lot of biomass, but it just doesn't make it into yield. I think it's worth remembering that partitioning is pretty important. For our cereal crops that were improved during the Green Revolution, this occurred largely as a consequence of greater partitioning of resources to yield. So it's something we really need to think about for our pulse crops. And that's because it's been noted in the literature previously that pulses still maintain a sort of evolutionary trait, I guess, to delay their seed production. So under drought, an increase in canopy biomass might not necessarily lead to an increase in yield. And this area hasn't been explored all that much, but results from the field, like RCN's agronomy studies, really show that it's an area worthy of a lot of further exploration. So maybe pulses have some kind of inherent sink limitation that sort of prevents partitioning to yield under particular conditions. And how might we explore this? Well, let's have a think about it. But first, some biology. So pulses are described as either indeterminate or determinate. And so indeterminate varieties are those that continue their vegetative growth through flowering. If you have any climbing beans growing in the backyard, you'll notice that they sort of keep climbing up and up and up, and they grow, they terminate their stems with a vegetative cluster, which also includes a flower. If you've got any barley growing in your backyard, my other example, you'll notice that its stems terminate in a flower cluster and vegetative growth terminates at flowering. So typically what we think about is that because of this longer period where flowering is occurring with vegetative growth, they have a longer flowering window and a longer period where indeterminate varieties are more vulnerable to stress conditions, compared to determinate varieties, which have a shorter flowering window and therefore a shorter period where they're more vulnerable to stress. And so it's often talked about the early maturity of these determinate varieties compared to indeterminate, but also there's a bit of discussion around the stability of yield for indeterminate varieties. And that's because there might be some sort of flexibility in their yield development in response particularly to stress conditions, because they're able to sort of compensate for any stress that might occur early on in the flowering period by developing yield a bit later. So all of our species are classified as either indeterminate or determinate, and pulses sort of sit along a bit of a spectrum with regards to their determinacy. And within species, varieties can also differ a little bit too. So common bean is a really nice example of this. Some varieties might also flip back into some level of indeterminacy under sort of stressful conditions. Seems like this is happening. So what's the critical window? Well, if we understand that window of flowering, if it's long or short for pulses a lot better, then we can really start to understand the period of time that they're going to be vulnerable to stress. And that's super important because that's influencing our yields. So this is just a hypothesized curve. There's a proposed reduction in yield potential occurring after a certain number of days of flowering when the plant is experiencing a stress. So drought or heat or frost, for example. And so if we can start to overlay information about the flowering window and the different stresses, then that is really going to be really useful to help us guide not just plant breeding, but also crop models and give farmers more information for their agronomic management practices as well. So to explore the role of timing a little further, when I was at CIAT, we set up a small-scale experiment in a lysimeter system, and we had 2 indeterminate varieties, and we noted and tracked the timing and location of all of the pods on the plant, and then we harvested each pod individually for weighing. So unsurprisingly, the pods that developed on the main stem, like you can see in the black here, they developed from youngest to oldest along the nodes. And the later developing pods clustered typically on the branches, like you can see in the open circles here. And that translated to differences in yields. So pods that developed later had a significantly lower seed weight compared to the pods that developed earlier. So in the blue is the earlier developing, the late is the grey, and this is the seed weight per pod. But that was only under well-watered conditions. Under water stress, those late developed pods had slightly lower, but not significantly lower yield. And in this experiment, under this particular drought scenario that we applied, the early pods seemed to be penalised. And that meant really that the plant missed out on developing these pods and diverted some resources to fill late pods. And then overall, the seed weight of the was declined under these drought stress plants. So during this study, I noticed that pod length was something that was interesting and matters and plays a pretty big role in common bean yields. And this is a nice contrasting example. So this is a really tiny little pod from a drought stress plant. It just has 1 seed inside compared to a beautiful long pod from a well-watered plant that has many, many seeds inside. So it's obviously something that's interesting. And the same was noticed in mung bean. So this is the mung bean NAM population, and there's also some commercial cultivars in here as well. And they were phenotyped in the field by DAF mung bean breeder Cole Douglas and his team as part of the ACR-funded International Mung Bean Improvement Network. So what you can see here is that for the mung bean varieties, pod length and 50 seed weight are nicely correlated. And these mung bean varieties are grouped by the size of their seeds and also their maturity. So the small seeded varieties are in blue, the medium are in green, and the larger in a nice coral colour. And you can see the different shapes are denoting different maturities. So early is a circle, late is a triangle, and regular is a square. And typically the early and regular maturing varieties and the large seeded sized varieties are up the top here. There's a little exception, and that's these 2 black grams, which are a different species, Vignamungo. They have very different architecture to green gram, but they're really interesting to look at in terms of their diversity and different traits. They are included in many mung bean studies. So seed weight and seed size are obviously related to yield, and you can see in this graph here. So these, again, these large seeded varieties cluster towards the top panel. And so what we really want are some quick indicators for how plants are responding in different environments, and potentially pod length or maybe pod growth rate might be good for us to have a look at. either way, there's obviously a lot of diversity in this NAM panel, and I'm really looking forward to studying this more with Cole and his team at DAF, as UQ are about to come on board as part of the Mung Bean Improvement Network, and we're going to dissect the physiology and the genetics of canopy development, and also look for a narrower flowering window for this population and for future mung bean varieties. So it's pretty exciting. So, as I mentioned, understanding the length of the critical window and its interactions with stress are super important. It's basically an essential first step, and that's going to guide many other targets, like the narrowing of the flowering window, synchronous flowering. That would have huge advantages in terms of management for abiotic stress for growers, but also for harvest for things like desiccation as well, which can be a challenge. I think increasing the overall sink strength of the plant is likely to lead to higher yields, and that could come from things like reductions in flower or pod abortion, which definitely occur when the plants are under more stress, but also by increasing the flower number or the pod number under ideal conditions, and also improving some of that partitioning of biomass to yield. And I think to effectively really understand partitioning, we have to develop some better indicators than just harvest index for pulse species. So pod length might be a nice quick indicator under stress conditions, but we need to do a bit more exploration around this. So the last area I wanted to touch on very briefly is nutritional quality. And pulses are important because of their high protein quality, but there's a bit of room for movement around this. So we know that stability of yield is super important. We want to know how any given genotype will respond in a particular environment, but we also need to know how these sorts of shifts impact on nutritional quality. So how does nutritional quality change across the resource availability spectrum? And in cereal species, this is relatively well known, but in pulses, we don't understand all that much at all. And I think particularly for a lot of these indeterminate varieties that have some flexibility in their yield development, it's something that we should consider. So while I was at CIAT, we undertook a field trial using some of those lines I showed you earlier that were adapted to phosphorus and drought. And we had a treatment under, well, basically an irrigated and a rain-fed field trial. And under drought conditions, we saw a massive reduction in yields, up to 70%. So we took samples from the plants, we took leaf samples and grain samples and sent these back and analysed them in the lab at Sydney for lots of different things, amino acids, micro and macronutrients, and sugars. And what we saw was super interesting. Well, I think it was interesting. So drought basically significantly increased the amino acid concentration in the leaf tissue. So this is a heat map. So basically, the darker the colour, the higher the concentration of whatever you're looking at. So in this case, it's amino acids that were detected in the soluble leaf tissue. And these lines here were grown under the rain-fed field trial, and these lines here were grown under the irrigated trial. And so drought significantly increased the concentration of amino acids in the leaf tissue here. And this makes sense because amino acids, well, some of them at least, have an important role in osmoregulation. So they're helping the plant basically to temper the effects of drought stress and maintain osmotic potential. In the grain though, we saw no significant difference in the concentration of amino acids, and that was pretty surprising to me. We know the plant is suffering from drought, and in other crops we know that protein content does change. And so there was no significant difference between either treatment for amino acid concentration. And we saw similar results for the micronutrients as well. There's no significant difference in the grain tissue in terms of the nutritional quality. But we looked at the soluble sugars in the seed, and this did change a bit. So myo-inositol, which I've just circled here, is a sugar alcohol. It's known to play an important role in osmoregulation in plants, helps the plant cope with drought stress. And it's obviously the reason why it's increased so dramatically here in the grain. So this is a problem in some ways though, because it's helping the plant cope, but for us it's not great because myo-inositol is a precursor to phytic acid, and this is the most abundant phosphorylated myo-inositol derivative. It's a known detrimental component of our foods. It inhibits digestion. It prevents the absorption of important nutrients like iron and zinc. And that's a big problem for crops like bean because people are trying to directly increase the iron and zinc concentration of these crops. So I think this is an area that deserves a bit of further examination to make sure that we're developing crops that are drought adapted and are nutrient rich. So what should we look at for nutrient quality? Well, for starters, we should at least consider it. If we're breeding pulses for human and animal nutrition, which we are in the majority of cases, we really can't ignore nutrients anymore. We have to measure this. And we should consider the interaction that's probably occurring between abiotic stress and anti-nutrients like phytic acid, which inhibit the digestibility and the absorption of other nutrients. And I didn't discuss this today, but for some pulses, protein content is fairly low compared to their other species, other pulse species. And so we could consider increasing protein content as well for some of our pulse crops. But we don't want to do that at the expense of yield. So in conclusion, some areas that I think are important for future research from a physiology perspective are understanding the optimal above and below ground architecture for stability and considering there's trade-offs that might be occurring between the above and below ground, the shoot and the root. Clearly a massive target is understanding the flowering window a lot more and considering whether it's possible to start to design pulse crops with a narrower flowering window that's stable or at least being able to manage through environmental fluctuations. What I also think would be super cool is if we develop some determinate varieties that have a cluster of pods at the top of the plant. This would make them much easier to harvest and look after, so I think that would be nice. We need to consider any trade-offs that are occurring between stress and anti-nutritional factors and also try and maximise our protein as well. And as for everything, we have to keep context at the front of our minds. Production scenarios vary and modelling these would be really useful to try and understand how things are occurring in different situations. So for the future, we can focus on things like high throughput phenotyping platforms to rapidly screen varieties for important traits, to inform things like maturity and guide management practices like desiccation. We really need to start to consider more the mechanisms and the relationships that really underlie a whole lot of traits for pulses, things like photosynthesis and partitioning. Exploring diverse germplasm is going to be critical to find varieties that have tolerance to abiotic stress, and especially for other traits that are important, like synchronous flowering, for example. And finally, we have to improve our crop growth models for pulses so we can further understand the implications of improving traits like synchronous flowering. Coupling photosynthesis into crop growth models like was done so successfully here at QAAFI for wheat and sorghum would also be really great for pulses, so we can start to see what would happen if we do look for things around photosynthetic capacity to our yields. So I have to finish by thanking many people and institutes that I've had the pleasure of working with and at, and that starts with RCN here at QAAFI and the rest of the Mung Bean physiology team who have been incredibly welcoming and wonderful to discuss ideas with. Cole Douglas and his team at DAF, thank you so much for everything so far, and I'm looking forward to working with you in the future. So I really loved my time working at ICRISAT and with the support from the Australian Endeavour Research Fellowship, and I can't forget my wonderful colleagues and friends at Sydney, CIAT and the University of Western Australia. Thank you. And so I'm happy to take any questions if you have any.
Okay, thanks, Millie. That was great. We have got a few questions already coming through, and so Miranda Mortlock has asked about the role of photoperiod and what role that plays in relation to the indeterminate nature of legumes and responses to drought. So are these things tied in? This could be a very complex answer, I would imagine.
Yes, the short answer is yes, they are tied in. The long answer is we probably don't know enough, I think, about this, but that's something that the Mung Bean Physiology Project is going to start exploring more, and Kylie Wennon's got some interesting data around this which I think is going to be really cool in the future because, yeah, it obviously has a big role to play in floral development and is something that's likely important and maybe why we're seeing differences across these environments across the state as well.
Okay. We've got a question from Kai. Kai is interested in the carbon isotope discrimination and he wants to know, is it scalable to breeding programs? Of course, he's thinking about using it on tens or hundreds of thousands of lines. Sure.
I mean, not the scale that you work at, Kai, but I think it definitely has been applied in the wheat breeding in Australia by people at CSIRO. But beyond that, I don't think it has been. It is difficult to take that many samples from that many plants and run them through a lab, basically. But yeah, it's possible, but it's not the most ideal thing now that we have these sorts of high throughput phenotyping platforms. We're much better able to deal with the numbers of populations that people like Kai work on.
Well, being a plant breeder, he's also asked, what's the heritability? Have people looked at heritability of this trait?
Yeah, I think so. I'm not sure about that in pulses though. And especially in chickpea, for example, one of the things I was looking at ICRISAT was whether we can actually use it for chickpea that well, because the correlations aren't so nice as they are in cereals. And that's probably because of things like the leaf acids that are accumulating on the surface of the leaf, potentially influencing the discrimination. So it has another fractionation event associated with leaf acids, and that might be influencing our signatures. So I actually, when I was at ICRISAT, took the leaf acids off the leaves before we dried them down, so we could make sure that this wasn't having an influence on our discrimination values. And yeah, I mean, RCN and others show this really nicely for peanut. It works very well for pulses, but for some like chickpea, potentially that's why we're not getting such clear correlations.
Okay, thank you. The questions are coming in thick and fast now. We're already up to 12. Not sure if we'll have time for all of them, but we'll do our best.
You have to eat lunch at some point, Ian.
Exactly. So Geetika has asked that in regards to the amino acid solubility, did you think the concentration was related to lower leaf area under rain-fed conditions?
Yeah, so that was on a gram per gram basis. So that accounted for any changes in leaf area. I don't think that was to do with leaf area. We did see differences in leaf area in another study in response to low phosphorus, and that had an influence on the drought response, but not in the field.
Okay, thank you. That's good. I think a lot of people are also have put in a few comments, and a lot of people, for some reason or other, people are saying they really like your talk. So that's good. That's good. Paulina has asked, do you think that drought limitation work is more important than nutrient limitation work in the context of pulses, especially considering nitrogen fixation?
Can't we have both? I mean, I guess that was what I was trying to get at, that we can't really think about one without the other if we're looking at systems holistically. And yeah, I mean, nitrogen fixation for pulses is a really interesting space. It obviously deserves a lot of attention. And has a big influence on things like protein quality. But I think that it's becoming more and more negligent of us to not consider the nutrition of the grain yields, especially with things like drought stress and the role of those sorts of deleterious compounds in foods.
Okay. Well,
I don't know if that answers the question, but.
No, I think that's good. You know, you can have both. That's the prerogative of young people to think they can have everything.
Yeah, that's right. Yeah.
I'm going to take that a little bit further and just say, so on deep soils, because you talked about legumes being largely shallow rooting, on deep soils, and we've got a lot of, like the Darling Downs, for example, it's deep soils. Is it effectively that the legumes are yield limited because of their inability to use nutrient and water at depth?
Yeah, not entirely clear. And I guess the thing that I often thought about a lot with common bean was that they're often grown on very poor soils as well. Like lots of legumes are just adapted to these sorts of marginal environments really. And so that's why they get into a soil with a little bit of nitrogen and they just don't even bother fixing any or developing any nodules. They become a bit lazy, I suppose. And maybe that's also something that we're seeing in the grain, is that it's just sort of this evolutionary requirement to germinate, and they don't put any other effort to getting any other nutrients in there. And the same with what's happening in the soil. There's no real requirement for them to get any deeper than they already are. They're going okay. So they don't need to look down into the profile. So it's changing that physiology that's come from domestication, I guess.
Okay, thank you. So I'm just going back to, so Milisu has asked, what have you done any work, or has anyone done work on the impact of chilling in chickpea? And is this something that you think is important for your research?
Yeah, I haven't done any work on frost. It's much more pleasant than heat, I suppose. But but there is a lot of work happening in that space in Australia. I haven't done any in that work, but it is 100% important. Has a big impact on yields, especially in Queensland. Yeah.
Okay. And one of the great disruptors in this area, a guy called Lee Hickey, has said that you've talked about roots, but what root trade is best to measure, and are there high throughput methods to phenotype pulse root systems?
Yeah, so root length density has been measured quite a lot for pulses. It's like the depth of the root system and how much biomass they have. And I guess that's a nice little over-encompassing one. There's obviously been a lot of work in common bean that's been done in root systems. Things like shovelomics were developed in common bean. I think roots are still the hardest thing to phenotype, and there's not really any nice quick way to do it. For any crop, let alone pulses, there's obviously indicators. We've tried ClearPot for chickpea earlier this year and it didn't work so well, but it'd be nice to keep trying that, I think. There's certain other measurements we can keep going, keep trying. But I guess there's also canopy indicators, and that might be our best bet for pulses. So looking at flyovers for drones and things, and canopy temperature and other traits that are nice proxies for how much water the plant has available to it.
Okay, and this one is probably more of a comment. It's from RCN, and he says sometimes synchronous and determinate flowering may not be advantageous. So, for example, if you get a pest attack during potting, it could be detrimental to yield.
Yeah.
So, and that makes sense. So we've got someone whose name is Anonymous, and this is anonymous attendee. Does chickpea have a more predominantly qualitative or quantitative response to photoperiod?
Oh, I'm not sure of that answer, to be honest. Haven't looked at that very closely for chickpea, sorry. Anonymous.
Okay.
I'll get back to you if you send me an email with your name.
Yeah, sure. And Yuhan is asking about legume species and says, do even legume trees have nodules?
Yeah, they do. Yeah. Legume trees are the coolest trees.
Oh, okay. Of course.
Obviously.
Of course.
Yeah, they're really interesting. Yeah.
I'll ask one last question, and it's Kai coming back for a second one. He wants to know, is there a lot of genetic variation for key adaptation traits among elite cultivars?
Yes, I think so. There is a fair bit, especially for some of the water use things that I've looked at for chickpea before, and it seems like there's a little bit there that we could play with. And yeah, I mean, looking forward to exploring more of that in mung bean, but it seems like there is a bit of variation there amongst those cultivars. There's probably not necessarily enough, and that's why we need to start expanding that variation into other more wilder relatives and developing interesting populations like Cole has for the NAM, because that allows for like a lot more phenotyping to happen and a lot more interesting stuff to come out than what we have currently in our elite germplasm.
Okay, and I'll take that further then to ask you about when you talked about wild germplasm. So Australia is home to species Cygna and Cajanus and glycine, etc. Do you think that's a place where it would be, I mean, it's always interesting to go and look at wild relatives, but do you think it would be a place where there'd be any low-hanging fruit that might be able to be introgressed?
Yeah, I mean, I think that's like a really nice place for us to, as Australians, to look at. We should be looking at this stuff a lot more than we currently are, and RCN's done some nice work on this for pigeon pea. And I think it's a really, yeah, exactly. It's a nice, easy target for us to look at these things, especially for crops like chickpea, where they come from just one little place in the world. It's nice for us to look at these other traits for Vigna. Yeah.
Okay, I had another question, if I may, and because I'm the chair, I guess I'm going to. You showed the stuff about cereal genetic gain, and you showed legume or pulse crop genetic gain, and we know that in some parts of the world there's been more genetic gain in cereals than in others. And it could be because of management, it could be because of genetics. To your knowledge, are there particular pulse species in particular places where there's been really large genetic gain, and are there lessons that we can learn from that in Australia?
Yeah, for sure. So, I mean, soybean and pea, even soybean particularly, but pea is a good example. It's going gangbusters in Canada and all these other parts of the world, and there's a lot of lessons we can learn from those crops and bring into our sorts of breeding programs that don't have as a longer time period in terms of the history. I think there's a lot we can do. Particularly soybean, they are so close to common bean. It's the same genetics, just doubled. So there's a lot we can learn from crops like soy, which have had a lot of time to improve over, that part of the US where there's so much soy grown. Yeah.
Okay. Well, thanks very much, Millie. We've just about run out of time, so we're going to bring it to an end now. And I'd like to thank all the participants for joining us. And
thank you.
It's a shame we can't all clap at the end, but I'll do it anyway, just to bring it across. So thanks very much, Millie. And if anyone wants to contact Millie, she's some of her time in the Hartley Teakle building. She spends some of her time up at Gatton. But it's easy to find her on the website, and she's always on email.
Yeah, just send me an email. Thank you.
Okay.